right, so just to recap for you where we've been this week, on Tuesday we concluded photosynthesis, and then we concluded Tuesday with aerobic respiration. We compared and contrasted aerobic respiration versus anaerobic respiration and fermentation. Where we're going in this journey today is we're going to look at better details of aerobic respiration. As you guys read ahead so that's not so traumatic today, because it's going to be somewhat similar to what we saw during the photosystems where electrons were used. And that's basically the same concept. So coming off of photosynthesis, the action of the mitochondria will be a little bit easier. It's not easy, but a little bit easier after coming off of photosynthesis. We learned that the whole point is we're going to slowly oxidize glucose to make what? Lots of sugars. I just repeat sometimes. We're not making sugar. What are we doing? We're oxidizing glucose to make ATP. Okay, so I just repeat sometimes. I shouldn't do that. In the mitochondria, we're going to oxidize or break down a sugar to make ATP. Okay, so we're going to do it real slowly so we can be real what? Efficient. We said the cell's going to do it slowly so that the production can be efficient. And yes, you will be very knowledgeable about this at the end of today, by Tuesday. We identified the primary phases of aerobic respiration, but we're assured to note that these weren't the only phases of aerobic respiration, but these are the kind of big key points. And so we summarized glycolysis. We said this happens where? In the cytoplasm, and it splits glucose or sugars into two pyruvate molecules and releases how much ATP? 2 molecules of ATP and how much NADH? 2 molecules of NADH. It splits sugar into. We'll get back. We'll repeat it in greater detail. We went through a nice review for about 20 minutes on yesterday. So glycolysis produces pyruvic acid is the answer, right? So pyruvic acid or pyruvate molecules. What we're going to learn is those pyruvic acid or pyruvate can be processed in an organelle called the mitochondria, and more specifically in the matrix of the mitochondria. We said pyruvic acid can get transported into the matrix of the mitochondria and start around something called the what? The Krebs cycle. Does the Krebs cycle produce a bunch of ATP? Not a bunch. What did it produce a bunch of? NADH and some FADH2. Very good. Thank you. FADH2. Tuesday. All right, so we'll come back. We'll revisit it. So we learned, right, briefly, the Krebs cycle produces a lot of NADH and FADH2, not so much ATP. So we said, well, the point of that is to produce the NADH and the FADH2, which we know do what? They carry electrons and, what's the H? Hydrogen ion. So we said we're going to use those in another step called what? The electron transport chain, okay, as the name implies. Okay. And the whole point of the electron transport chain is to transport electrons to, it takes electrons from NADH and FADH2 to make ATP, and a whole bunch of ATP is made because during the electron transport chain, you're going to see that similar molecule called what? That can make ATP synthase, right? ATP synthase. At the end of the electron transport chain, ATP synthase is going to synthesize lots of ATP. Okay. That was the review that we covered on Tuesday, so now we'll get into more details. So if you weren't here on Tuesday, Don't completely panic or flip out yet. We'll add the detail. We'll repeat it more specifically. Do what? So this is about where we left off. We said in order to metabolize, we're going to follow this narrow pathway for the catabolism of a molecule of glucose. And what we summarized, do we need a diet only of sugar? No. What else we said? We also need to eat proteins and fats and other lipids. Okay? So in this class, we're going to follow, in this image, the middle pathway where sugar is catabolized. You see the green highlighted pathway. We're going to start with glycolysis, produce pyruvic acid, okay, and then drive the Krebs cycle, and then the electron transport chain. So we're going to go along the middle. However, other molecules, nutrient molecules, can also be catabolized to power the Krebs cycle and power the what? The electron transport chain. So, if you understand glycolysis and where it originates and how everything happens after it, you have an idea. You don't know the details, but you can understand, well, amino acids can be broken down into what? Pyruvic acid, right, which could start a Krebs cycle, right? Lipids can be broken into a molecule that's a byproduct of pyruvic acid metabolism, which could then start a full cycle of the Krebs cycle. And okay, so the point is, sugars are not the only nutrient source, which you know. Okay. Let's look at glycolysis. Okay, so I've got a kind of open room for you on page 2 to kind of just take notes. We summarized it, but we'll go through it a little bit slower to understand it a little bit better. The name glycolysis, lysis means what? To break down, to split, to break. What are we breaking? Sugar. Glycolysis is a process in which we are breaking sugar. The sugar that we are going to study is glucose. Okay? So we're going to catabolize. Remember that word. What does that mean? Catabolize. Process. I could just as well say oxidize, which means we're going to take off its electrons. So there's a lot of ways that you can say this. Glucose is oxidized. Glucose is catabolized. Glucose is lysed. So we're breaking it down to do what? To release its energy, okay, and make ATP. Question is, where does the glucose come from? Well, it's got to get into the cell. And glucose is a large molecule. And so, think about an organism. The nutrients would be on the out. Think about yourself. When you eat something, the food goes in your stomach, and then that gets broken down by the digestive system and goes into your blood, right? You have a meal, your blood sugar goes up, and the nutrients go to the blood. And then those nutrients need to go from the blood into your individual cells, like your skeletal muscle cells or the cells in your brain or wherever. And so the glucose has to get inside of a cell for it to be used. Does that make sense? Okay. The way it gets in there is through facilitated diffusion. Okay, so it's just facilitated across the membrane. As soon as glucose is transported into the into the cytoplasm of the cell needs energy. It's going to immediately be catabolized. The enzymes are available. It's going to immediately be processed. Substrates available. Now we can process and produce our product. However, when this process starts, we actually use 2 molecules of ATP. Like, aren't we supposed to be making ATP? Here we're using it. So we're actually in a negative energy balance when glycolysis is initiated. also summarized earlier that is, well, glucose has how many carbon atoms in it? 6. We split it, we're going to produce 2 3-carbon containing molecules, 2 separate things that have 3 carbons in them. So it's going to be split right into half, and you might want to identify that those, we'll give them a name in just a second. It's going to be split in half. And Miss Lori, I've changed the colors again, so I'm going to test in the back. Is this gray too light also? Okay, thank you. So glucose is split in half. We'll give the name in just a minute. And along the pathway, 4 ATP molecules are produced. So I used 2. I made 4. What's the balance? 2. Okay. So there's a net production of 2 molecules of ATP following glycolysis. It's in the cytoplasm. It's already there. The cell's always metabolizing, so they're available. The glucose has to get ready, and the way it gets ready is it gets phosphorylated. And so everything's just, you can list all the billions of molecules that are hanging around, and in this case, ATP is used. So it takes energy to get the molecule ready. A very unrelated visual, okay, but maybe it'll help you. Like if you want to eat, you have to chew, right? And that takes energy, right? You're like, well, where does that come from? So it takes energy to transform the molecule to get ready.
So if it takes energy, does that mean that like pull up it's vital way to an enzymatic reaction?
It is at first. It's a series. It's a metabolic pathway. And if you remember, our metabolic pathway is one thing, then another, then another. And so in the first couple of reactions, they are endergonic when we're using ATP. But it's the last 2/3 of the reaction that are exergonic. When E is good observation. So the thing is like, well, what's the overall picture? The overall picture is ATP is actually made in a net balance of how many? 2 molecules. And that's a lot of times when you take these classes, people hone in like 2 ATP. That's important, but that's not the big deal. What's the big deal with glycolysis? Splitting the sugar to form, what is that carbon-containing molecule? Pyruvate. And also, what else is given off? What do we really care about? Yes, ATP is useful. We can use it right away to do another round of glycolysis, but the carbon dioxide is not liberated yet in this process. NADH. Excellent. Okay. NADH is produced. And we know this is very similar to NADPH because it's carrying for us. We picked up some electrons and hydrogen ions. We're going to use these where? What could you write about NADH? These will be used probably where? In the electron transport chain. Very good.
So the 2 net, is that the 2 NADH?
The 2 net, yeah, it's for ATP. So if I just think about your bank account, let's say you got $0 and you spend 2, so you're at -2, and you put in 4. You don't have $4, you have $2. Okay, so the 2 net is, and that star is to the ATP being
We
produced.
had 2, we made 4, we had 2 left, and that's what fell down there. That's what the 2 NADH is.
No, ATP is a different molecule than, so completely unrelated. So the net production is in reference to just ATP.
Okay.
NADH is a completely different molecule. It happens to have the same number of 2 that's being produced. Is maybe where you're getting tangled up. So 2 ATP, how much NADH? 2 molecules. So I could use the word net. Net just means overall. I could use the word net for NADH as well.
Is that what we're going to, like, really have to know for the test, the net?
Yes. 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 So following glycolysis, there are 2 molecules of ATP produce 2 molecules of NADH, which could be used later in the electron transport chain. But the big payload is not the ATP, not the NADH right away, but what? The pyruvic acid. I keep saying pyruvic acid, which is another name for pyruvate, and I'm confident that your book uses, did you use pyruvate? Okay, so I'll try to just change my notes. Two pyruvate molecules are produced. These are the glucose that was split. Okay? When it's split in half, it's called pyruvate. So maybe you want to note that pyruvate are the molecules that have 3 carbon atoms in them. The pyruvate is the two separate 3 carbon containing molecules that we described in here when glucose is split. When glucose is split in half, the final products are called pyruvate. Yes. I'm sorry? The 6 carbon glucose. We got 3 people talking at the same time. Miss Kinchin.
Okay, I think you also say the same thing, like, okay, the pyruvate, when you said 2 right there, it's telling you I have 2 of them. There's 3 in each.
Yes, yes. Miss Bennett?
That's it.
Okay. So it was a 6 carbon something. We broke it in half. It's no longer glucose. These things are called pyruvate. This next image that I have, do not have for you in your handout, is just there for me a prompt to remind you that glucose is transported, facilitated into a cell. So if you look at this image, the outer purple is representing a cell membrane. This could be a plant cell. This could be a cell in your brain. This could be a cell in your dog's tail. Whatever. It's going to have a membrane, and if it needs glucose, it's going to have a glucose transporter. Okay, so it gets transported into the cytoplasm, where the describe the glycolytic pathway or glycolysis occurs. So you see this thing right here, the little purple thing, is the glucose transporter. Those reactions occur in the cytoplasm. Under ideal, before I move on, is oxygen used in this pathway? I didn't show you the pathway. Let me see. All right. Do you see up here in the figure 6.2 in the overview, where glycolysis is in the green box? If something's being used and an arrow goes to it, and something's being produced and arrow's coming out. For just the glycolytic pathway, is oxygen used? In just the cytoplasm, do we see oxygen input? No. So glycolysis is what type of pathway? Anaerobic. Write down on your notes that glycolysis is an anaerobic process. Does that imply that the little cell that's using glucose is about to keel over? It should be on your first page too. ANAEROBIC. What did we say? If the pathway may be anaerobic, but what? What was the big point I was trying to make on Tuesday? But what? It's not using oxygen to pick up electrons, but it doesn't indicate what? Doesn't indicate that oxygen is present or not. Oxygen it better be present in a, like say for your cell or a plant cell. It's going to be present. It's just not used in this reaction. It better be diffusing into that cell. It's just not used for this reaction. So So the glycolytic pathway, or glycolysis, is definitely, hands down, undebatably, right, anaerobic. All right, so we continue. What we said is the whole point of glycolysis, it is a slow oxidation of of glucose. We said to be efficient, and if you remember with chemical reactions, they are exothermic, they can release heat, okay? And so we're releasing heat, so just metabolizing nutrients keeps cell temperature normal. But we want to do this slowly to be efficient for obvious reasons, but also if we were to release all of the bonds in glucose at the same time, so much heat would release that what? The thing would just heat up and die. Okay, so there's so much potential energy in a single molecule of glucose. If we don't do this slowly, cells may become too hot and lethal, and then it's completely pointless. So we do it to be efficient and to keep the temperature at normal set point for that organism. We also identified that this process does occur under anaerobic conditions. We said has no implication of presence or absence of oxygen. It just says oxygen's not used in this process. I want to refresh for you the role of these carriers. NAD, if you remember, it's very similar to what? NADP, okay? And it can, right, in this state is oxidized, but it can pick up what? Electrons from what? Hydrogen, okay? And so NAD and two hydrogen atoms, right, can interact, but what we notice, like for example, in NADP, it's partially charged, so it cannot accept the full hydrogen atoms. And so when NAD is reduced, it forms which molecule? NADH, okay? And also a free hydrogen ion. So this part should seem pretty familiar. This molecule just doesn't have an extra phosphate, like what we saw in the chloroplast. So it's just a reduction, oxidation reaction. Notice how the arrow is reversible. Remember, we can form NADH, or NADH can give up hydrogen ions and electrons to form NAD. We just said it's a carrier. So our first step that we see NADH being produced is here, following glycolysis. Are you all okay with the role of NADH? It's similar, just transporting hydrogens and electrons. And so figure 6.4 might be a good point for you to go to and kind of review what happens. This is zooming in a cell or right around a given mitochondria. It would be ideal if they had an entire cell membrane so that you knew where you're at. But the glycolytic pathway, you know, occurs where? In a cell's cytoplasm. Okay. Does or does not use oxygen? Does not. And maybe it's the next image, somewhere around there in chapter 6, goes into more specific detail. Obviously, I'm not going to have you understand or draw the chemical formulas or anything, but if you can understand the features, which I've been repeating since Tuesday, inputs, outputs, byproducts, okay, location.
Do hydrogen ions are somehow produced in glycolysis? Does that mean water is the byproduct?
Water is consumed in this pathway. I don't think water is liberated in the last step. So yeah, step number 9. So yeah. Okay. You gotta go to the dark side of the biology major. Putting everything together so quickly. Good job. So there are 10 reactions in the glycolytic pathway. Okay? And there are some hydrolysis reactions going on, and we can see some NADH and ATP being liberated. But if you go through, when you review this image, you'll never see oxygen entering. Actually, we even see carbon dioxide exiting this pathway either. So no gas is used or produced in the glycolytic pathway. All right. Let me show a quick animation. This animation is going to go into much better detail. I think it goes into the names of everything. But if we look at the overall respiration, aerobic respiration, we're going to start out with the glycolytic pathway, and at the very end will be the events of the electron transport chain. So we'll go there in a minute. But if we specifically look at glycolysis and producing pyruvate, we have a 3-carbon molecule transported into the cell cytoplasm, and what we noted is that it's an expensive process, in that we actually spend 2 molecules of ATP priming, and that you can see how it's kind of changing shape. So we actually go in the balance, negative balance of ATP when we start out the glycolytic pathway. Okay. Just getting it ready. And then it's split. It's not immediately pyruvic acid. It's got to get further processed to become pyruvic acid. But notice that there's 2 molecules now that will each individually be processed. NADH is released. We know that can be used later. So it's going off to do its work. And now we first see our 2 molecules of ATP being produced. So we're at balance of 0 for overall net ATP synthesis. Gets rearranged to what's called isomers, and then we can produce 2 more molecules of ATP. At the end, there are 2 pyruvic acid molecules shown. This is a pointing device, not showing that they're joined together, that little Y. So, and this certainly is not structurally correct, but you can see there are 2 separate 3-carbon molecules called pyruvic acid, or pyruvate. This is not the end of the story. Those pyruvic acid molecules have more potential energy in them, and therefore can be further processed, or catabolized, or oxidized. Your book doesn't go into great detail illustrating what I'm going to call the intermediate step. So when you go in your book and you read the KRB cycle, that first paragraph, it doesn't label it, it just says this happens. It doesn't give you all the detail, but I think the detail is important. When oxygen is available, pyruvic acid or pyruvate will be transported into the what? Into the matrix of the mitochondria. Here's the big key right here. I want you to put the big if and underline it. If things are good and oxygen's available, then this will happen. Then each pyruvate molecule will be transported into the mitochondrial matrix. That's a big if. Okay, if it's not, something else completely different happens. So pyruvic acid is produced in everything hunky-dory inside of the cell. Oxygen's available. The pyruvic acid is actually transported across the outer and the inner membrane of the mitochondria and localized in the matrix of the mitochondria, where they will each be further processed. Okay. The pyruvic acid molecules will be processed, and since we're messing with them, they're turning into new molecules. And the new molecules that they're transformed to is called acetyl-CoA, acetyl coenzyme A. This, what I'm referring to as the intermediate step, also does not require oxygen to occur. It doesn't require oxygen in its pathway, which seems contradicting to what I just said. if oxygen is available, the pyruvic acid is taken into the matrix. However, oxygen is not used in this pathway. In this specific intermediate step to make acetyl-CoA, oxygen is not an input. When acetyl-CoA is produced during the intermediate step, we get a couple more molecules of NADH. That's good. Those can be used in the electron transport chain. And this is where we first see a release of carbon dioxide. Yes. Yeah, so the input would be pyruvate. The final product in this pathway would be acetyl-CoA. The byproducts would be NADH and carbon dioxide. I also need to be clear on something. The numbers here, it says each pyruvic molecule is transported in. So I'm saying net for both pyruvic acid, 2 NADH and 2 carbon dioxide are released. So if I were to look at it for each pyruvic acid, how much NADH is made? 1. And how much carbon dioxide is released? 1. How much acetyl-CoA? 1. Each one is converted to a single molecule of acetyl-CoA.
I have a question.
Yes.
So in the intermediate step, no ATP is actually
No ATP is produced during the intermediate step, and that right there is why it's often just overlooked. No ATP is produced. No oxygen is consumed. So some people just say, no big deal. But it is a big deal because it makes for us acetyl-CoA. Okay. Additional questions? Where is this happening? In the matrix of the mitochondria. It'll only happen if what? Oxygen is available. Does it itself use oxygen? No. It produces for us carbon dioxide, NADH, and acetyl-CoA. The big star of this reaction is the acetyl-CoA. That's what we want to focus on. Because it has more bonds and it has more we have bonds, we are storing energy, okay? So we want to pick off more of its potential energy, okay? So the intermediate step is just that first paragraph in the Krebs cycle. It is not, make a note, this is not the Krebs cycle. What happens before the Krebs cycle? I don't want you to get confused when you read your book. It is not the Krebs cycle. It happens before the Krebs cycle. We're to look at it from your textbook image in the figure 6.5. If oxygen is available, pyruvic acid is transported across both the outer and the inner membranes and processed in the matrix. And first, the intermediate step happens so that acetyl-CoA is produced, as well as some carbon dioxide and NADH. That's the intermediate step. It looks like your book calls it the transition to the mitochondria. So whatever, intermediate step. Some people call it the linking step. Whatever. It's making what? Acetyl-CoA. Okay. Is it making ATP? No. Is it using oxygen? Good. No. No. All right. So here's the key to initiate the next round of processing is the production of acetyl-CoA. Acetyl-CoA, once available, is what begins the Krebs cycle. And that's to get a full spin of the Krebs cycle. Acetyl-CoA is what initiates it. So notice the Krebs cycle processes a single molecule of acetyl-CoA. So if we think about it, really, how many molecules of acetyl-CoA were produced? 2. So really, the Krebs cycle happens twice if we go back in time to looking at a single molecule of glucose. So the Krebs cycle will happen twice under ideal conditions. Does anybody know where this happens? Actually, still in the matrix of the mitochondria. We're still hanging around in the matrix. That acetyl-CoA is in the matrix, and enzymes are there ready to process it, to drive. You can call it the citric acid cycle, you can call it the Krebs cycle, the tricarboxylic acid cycle, another one of those things that has multiple names. But it's a series of reactions that do occur in the mitochondrial matrix. Okay? What this does is it processes the acetyl-CoA and then produces something called citric acid, and that's why there's that alternative name for this pathway, the citric acid cycle. This reaction is strictly also anaerobic, so it does not use oxygen directly. This Krebs cycle is a metabolic pathway, just like what we learned before. One substrate will be the product, okay, and then serve as the substrate for the next enzyme. So we just process it step by step. So again, we're not going to go through each steps, but we do know acetyl-CoA starts it, and we know that citric acid is produced. What we're going to focus on are the byproducts following the Krebs cycle. And as we summarized on Tuesday and this morning, what is produced following the Krebs cycle? Which molecules? You don't have to do the numbers. What? ATP. What else? FADH2. NADH. Carbon dioxide. Very good. Now, I'm going to have two numbers up here. I'm going to put a value, and then I'm going to put a value behind it in parentheses. The first value is per turn of the Krebs cycle, but the second parentheses is, well, we know the Krebs cycle happens twice under ideal conditions, so the second number is doubling if the Krebs cycle happens twice. Does that make sense? The first value is just per directly from each Krebs cycle, or the second value is just like, well, let's presume under it happens twice, so we're doubling everything up. And so if we look at the numbers, each turn of the Krebs cycle only produces one molecule of ATP. So it still isn't very abundant ATP-wise. However, right, so if each spin of the cycle makes one ATP, if the cycle happens twice, we can make 2 molecules of ATP. So it doesn't look very exciting for ATP at this point. Carbon dioxide is liberated. So 2 per turn or 4 collectively. We also noted that NADH is produced. How much per turn? 3 molecules per turn of the Krebs cycle, or if we say it happens twice, 6 molecules of NADH. That's big time, because those NADH we know are carrying electrons that we're going to use in a minute. So we're getting a lot of electron carrying ability. And per turn, a single molecule of FADH2, or collectively, how much? 2 molecules of FADH2. So I just want to take a minute to introduce, or I think I mentioned it, but reintroduce to you what FAD is. It's very similar to what? NAD. Okay. And so what happens is it can also be reduced or oxidized. Notice the charge of FAD is what? Neutral. So when exposed to 2 hydrogen atoms, it can actually accept the entire hydrogen atom so that it produces FADH2. It does not liberate any free hydrogen ions out into solution. But it accepts the entire hydrogen atom. Basically, F80H2 is an electron hydrogen ion carrier. So up till now, how much ATP have we made? In glycolysis, we made 2. Intermediate step, we made 0. Krebs cycle, we made 2. Okay. So we made how much ATP? 4 molecules. Go back and look at it. It costs 2 just to get glucose ready. So it's not looking too significant at this point. And so we need to get to a step where we're going to see a payload. And the payload is in this process called the electron transport chain. And I think I have an animation. So before I get into better detail on the electron transport chain, let me show you this animation for a Krebs cycle. Alright, so just to put it in perspective, during glycolysis you made pyruvic acid, and then if oxygen is available, the pyruvic acid is transported in mitochondria and converted into acetyl-CoA. Then the Krebs cycle can initiate. That's where we just finished, NADH, F80H2, ATP, and releasing carbon dioxide out in the atmosphere. Or if it's a plant, the plant can use it. Okay? Now we're going to see, but actually the animation we're going to see is this Krebs cycle, and then we'll follow up with the electron transport chain. Alright, so here's our little mitochondria. So it looks like it's just diffusing. It's transported in. There are pores in the outer and the inner membrane to physically get pyruvic acid into the matrix. Okay? The molecule is decarboxylated, or you can see it losing carbon. Yeah. So we've got our little intermediate step going with a production of acetyl-CoA, and we get some NADH released during this intermediate step. Slow down. Alright, so to start the Krebs cycle, that molecule, acetyl-CoA, this little molecule right here, is added to how many carbons? It has 2 carbons, and it's added to a 4 carbon, so we're getting a 6-carbon molecule that we're going to process. Okay? That forms what's called citric acid. Okay? The citric acid is then processed. We don't need to get into the details. We'll see it processed. But what we do care about is NADH being produced. We do see some more carbon dioxide being liberated in the Krebs cycle. More NADH. You see that? I like this little animation. It's got the hydrogen ions and the electrons, and what we're going to deal with later. More decarboxylation. We'll see some ATP being synthesized. That cell can use that right away. And then F80H2. Notice how it's picking up 2 hydrogen complete atoms to form F80H2. And a little bit more processing, and we get another molecule of NADH. Alright. And then it's called a cycle, just like we've learned before. The end product is what starts a new round of the Krebs cycle. What questions do you have about the Krebs cycle at this point?
So all it does is process the acetyl-CoA.
Acetyl-CoA. That's what it does. It processes acetyl-CoA to pick off efficiently and slowly as well. What are they picking off? Electrons and hydrogen. then what would happen is the next step and the final step, processing a molecule, original molecule of glucose, is what's called the electron transport chain. This is where we'll put into practice the use of NADH and FADH2. The actions are very similar to what you saw following the light-dependent reactions in the chloroplast. Don't confuse that statement right there. I'm just saying the process is similar, but we are not in the chloroplast here. What organelle are we in? The mitochondria. Okay, we're in the mitochondria, so completely different organelle. Okay, but what I'm saying is it's similar. Is we're going to see a hydrogen ion gradient created, so we can use ATP synthase to produce ATP. Okay? So we'll see some similarities in that fashion. So the electron transport chain is going to harvest electrons from NAD and FADH2. The mechanics are very detailed. If you can just tell me that and understand that the electrons come from NADH and FADH2, I'm good with that. We're not going to go through and name all the little proteins in the order of reactions, but we know that the electron transport chain is using electrons that originate from where? They come from the KRB cycle and even glycolysis, and most specifically, which molecules from those? NADH and FADH2. Very good. So it's going to use the electrons from NADH and FADH2. What this does is create a hydrogen ion gradient. Okay? And the gradient, the directionality of the gradient is important for us to understand. The gradient is established so that hydrogen ions are in greater concentration in the intermembrane space. Now remember where that is? I-N-T, the spelling is important, I-N-T-E-R, intermembrane space. Okay, they're greater in the intermembrane space and lesser in the mitochondrial matrix. You said that word, E-N-T-E-R? I-N-T-E-R. Oh, I-N-T-E-R. I'm sorry if this is light. It looks, the colors look good on the, when I put them together, but the projector makes them look lighter, so I'm sorry if you guys are having trouble with the coloration. The what? The hydrogen ions are in greater concentration in the intermembrane space and lesser in the what? In the matrix. The whole point of creating a gradient, so I'll repeat it, so the whole point of creating a gradient is to transfer electrons. The electrons are used to create the gradient, so what's the point of creating a hydrogen ion gradient? We mentioned it a minute ago, I just want to repeat it. To use ATP synthase to make ATP. We're going to create a gradient to use it. The electrons are used for their energy. Remember, so the electrons have potential energy in them. The electrons provide energy to the little hydrogen ion pumps that pump hydrogen ions from the matrix to the where? To the intermembrane space. So those electrons help create the gradient. The whole point is once that gradient is produced, it can be used by a molecule called, just repeating, ATP synthase. So following the electron transport chain, we know what is produced. There's a molecule that gives itself away. What's made following the electron transport chain? ATP is definitely made. Okay? And I'll give you guys some numbers in a minute. From your handout that you guys turned in on Tuesday, what else is released following the electron transport chain besides ATP? What is another byproduct? What'd you say? No, that's always in there doing its own little thing. byproduct because what important molecule is used here that can combine with hydrogen ions to make water? Oxygen. This is where oxygen is used. So hands down, the electron transport chain is aerobic. Hands down, aerobic respiration. It's complex. We're just going to try to get the meat and potatoes from this process. But oxygen is used to do what? Was that if it's aerobic respiration, oxygen is used to? The electrons, okay, to accept electrons. It will then bond with hydrogen to form H2O, water. So oxygen is used to accept electrons after they've done their work of creating a hydrogen ion gradient. So again, the oxygen is used to accept electrons after they've done their work of helping create that hydrogen ion gradient. We also know that ATP synthase is involved to make what? ATP, I've kind of got this out of order a little bit, but water is produced and ATP is produced. Pictures will help a lot more. So let's go ahead and look at the image from your textbook, figure 6.7. Notice that we're really zooming in to a mitochondria. We can see the outer membrane, we can see the inner membrane folding, and we can see the intermembrane. It says compartment, space, same. Okay. The proteins that drive the electron transport chain are colored what in this image? The purple structures, okay? As well as is ATP synthase. Okay?
So that works the same as the one for photosynthesis?
It works similarly. The direction is kind of opposite. Okay? But yes, it uses the current, the hydrogen ion current to rotate and produce ATP synthase, or ATP, excuse me. So it works exactly the same. The directionality where ATP is released is opposite, but same concept. Good. What happens is the electrons, you can see they come from, they're here in the purple dots. They originate being carried by NADH and FADH2. The electrons are used to give energy to the proteins to pump what? What are we pumping? Hydrogen ions into the what? Intermembrane space. We're creating a gradient. That's only going to work if electrons are there to give power to it. Okay. Once the hydrogen ion gradient is established, it can be used by a completely different protein called ATP synthase, which rotates and produces ATP. So this is the whole thing, and I'll give you some numbers in a minute. It produces a lot of hydrogen ion, or excuse me, a lot of ATP. But if we follow the electrons, they have to go somewhere. So what can accept them? Oxygen, making this process aerobic, and because there are hydrogen ions around, it can couple with hydrogen ions and form water. That's metabolism. It's 30 minutes right there. A semester or more to really understand it, but we're kind of getting the high points, right? Hopefully we're getting the high points. Let me show you this animation. It might help a little bit to see it in practice. we're kind of finishing up the end of the pathway. Okay? NADH and FADH2 are going to be used to drive this process called the electron transport chain. This is occurring where? Where are we? In the inner membrane of the mitochondria. And so we can see our little transport proteins. In this image, they're blue. ATP synthase is this little funny looking thing on the far right with the little channel. Okay? What happens is NADH will donate its electrons, and it's going not at the right ratio, but when the electrons are donated, they kind of got it out of order. It would be best if they did the electrons, then the hydrogen ions flowing through. You felt like the electrons are what give the energy to pump that across. Okay? Then the electrons are transferred to another protein which gets more energy. Okay, gets more the electrons give more energy, okay, to pump more, and this one's showing FADH2. We can pump more what? Hydrogen ions, so to create more of a gradient. The electrons can then get shuttled to another protein that gets more energy to do more pumping. Okay, this one hydrogen ion pumping. You can see all the hydrogen ions being pumped across, creating a gradient. They may come from NAD. They may just be free in the cytoplasm. They don't have to come from NAD. They're everywhere, and so they may happen to be directly from NADH. They may not be. If it's easier for you, just think that they do. It might be easier to say yes. That would be a false statement. If it's easy for you, you're not going to go into cell biology, I'm going to guess. So if it's easier for you to say they do come from NADH, that's fine. At the end of the chain, though, the electrons have to go somewhere. We can't just accumulate all these electrons. They've got to be accepted by a molecule called. The electrons are accepted by oxygen. Oxygen is our electron acceptor, which makes this process aerobic. Okay. Let's see oxygen come in and pick up these. Come on, oxygen. There you go. So oxygen, can you see it picking up the electrons? Okay. And then because there's hydrogen ions around, they're nice and stable and can form water. All right? That's what the electrons do. But really, we kind of care about that gradient created by this process. And so at the end of the process is the role of the ATP synthase, which lets hydrogen ions flow through it from where? From where? From the intermembrane space through ATP synthase into the what? Back into the matrix. They just go kind of round and round. Okay. But the point is that once they go back through, that gives potential energy to make a lot of ATP. Look at the number over here. This is including everything from glycolysis and the Krebs cycle. But we know those produce 4. So the electron transport chain can make about how much ATP under ideal conditions? Just the electron transport chain can make 32. We said the 36 has glycolysis, and that 36 is including the 2 from glycolysis and the Krebs cycle. We take those out. Under ideal conditions, the electron transport chain can maximally produce 32 molecules of ATP. You look at it at the end, that's way more abundant than 1 or 2, right? 3 times more, right? So certainly, electron transport chain is a much more efficient route for ATP synthesis. Okay, so aerobic respiration is the preferred route for maximal ATP synthesis in a eukaryotic organism. So I want to make sure we can kind of see where the numbers are coming from. So I want to summarize this process for you. So at the end of your handout, you should have a nice little summary table, okay, on page 4 of your handout. This will summarize, this is what we really quickly said on Tuesday, what I really quickly said this morning, and then we'll summarize today with. So this summarizes about 2 hours worth of talk right here. Okay, so this would be something that you might want to quiz yourself on to see if you could really understand it. You know, maybe fill it in the chart on your own or write it in another essay, and so you can put the whole picture together. So, under aerobic respiration, we start out with a single molecule of glucose. What is the first step? What is the first step? Glycolysis, okay, is the first step that we're going to process. What your hand is, it looks like you probably didn't turn out. So how much ATP does glycolysis produce for us? 2 net molecules of ATP. And this, let's go through this. All right. We're working down. All right, so glycolysis is one step. The next step is intermediate step. Then the next step is Krebs cycle. Then the electron transport chain. Bless you. The glycolysis produces for us, you said, net 2 molecules of ATP. I think I'm working down. I can't remember. Intermediate step produces 0 molecules of ATP. Krebs cycle, net production of ATP is 2 molecules of ATP. And the electron transport chain maximally can make 32. There's always going to be a variation, but you can say no more than 32 under ideal conditions. So I got the little about 32. It might be, not everything's ideal. But under exact, precise conditions, you make about 32 molecules. That's the max. Yes. Location-wise, glycolytic pathway occurs in the cytoplasm, cytosol, those are same terms. What about the intermediate step? In the matrix of the mitochondria, make sure you identify that it's in the mitochondria. Matrix is just a word that describes a movie, right? Right, so this doesn't occur in the movie, it occurs inside of the matrix of the mitochondria. Okay? Krebs cycle occurs also in the matrix of the mitochondria. What about the electron transport chain? Where does it take place? Across that inner membrane space of the mitochondria. Across the inner membrane of the mitochondria. Regarding glycolysis, besides ATP, what else was produced? 2 pyruvic acid molecules. That's what we want to hone in on, is the pyruvic acid, and what else? What else was made? NADH, and also some hydrogen ions. I didn't really focus on those today, but they're liberated. Someone said, well, where do they come from? And yes. Intermediate step makes for us a molecule we care about called acetyl-CoA. It does make NADH and also releases carbon dioxide. Okay? So we care about the acetyl-CoA, but carbon dioxide is liberated. If it's in your body, it goes out into your blood, and in your lungs, in the atmosphere. The plant, it may diffuse on over to what? Into the what? A cycle, and the chloroplast called the Calvin cycle. There you go.
It was a C, but it just wasn't counting.
So it gets out of the mitochondria. Okay. After the intermediate step produces acetyl-CoA that can drive the Krebs cycle, which produces for us ATP. And most importantly, FADH2 and lots of NADH, and also releases carbon dioxide. And then NADH and FADH2 are used in the electron transport chain, which produces a whole heck of a bunch of ATP, but also what byproduct is produced here? Water, H2O. All right, I will not hold you accountable for the steps of fermentation. However, you know what fundamentally fermentation and anaerobic respiration are. And so you're also fully aware that not just sugar is utilized. And so for your exam, we'll cover all of Chapter 4, all of Chapter 5, and through 6.7. Again, it talks about other molecules being used. Okay? I will not hold you accountable for the specifics of 6.8. If you look in your handout, there are some nice supplemental videos, okay, that same virtual cell site that I gave you guys last week. And there's also a nice summary table or image showing the relationship between photosynthesis, Chapter 5, and aerobic respiration, Chapter 6. I would work on these, reprint them, and practice. And so we'll have an exam on Tuesday. So have a good, safe weekend. And if you need help with material, just shoot me an email. I'll answer as best I can.